Hello, am I audible? Hello. Yeah. Hi. Can you tell me what is your background? Okay. From which university? Mumbai University. Okay. So KC College in Mumbai only, right? Yeah. We'll wait for just 5 more minutes and then we'll start. Okay, I will start. So, in the last class, what we discussed was about how what enzymes are made up of. That is, mostly enzymes are protein, and which constitutes the primary structures, secondary structure, and tertiary structures of protein. And then we also studied how to analyze these primary, secondary, and tertiary structure of proteins. In today's class, the focus will be on how to get these enzymes, how to get this purified enzymes, and how can we use these enzymes for study. Okay, so let's begin. So we need enzyme in order to study. So the enzyme can be produced in two different forms. Okay, so first is where we can purify it. Okay, from directly from the natural source. Okay, we can purify it from the natural source. Um, We can directly purify enzyme from natural sources, or the second step is we can overexpress a particular enzyme. Directly purifying it from a natural source has many limitations, as in we we might not get that abundant or large or excess amount. We not might not get excess amount of enzyme. That's why very limited studies are only possible with such small fraction of enzyme, and we need to do multiple isolation, okay, from without overexpressing enzymes. So there are two methods: either we overexpress or we don't overexpress the enzyme. So and if we don't overexpress the enzyme, we need to isolate this enzyme from the natural source, uh, like for multiple times. And because of that, that's the limitation for to use enzyme without overexpressing it. But if we overexpress enzyme, we'll have large amount of enzyme which could be used for different analysis or other downstream application. So this is the scheme for producing enzyme, which by overexpressing the enzyme. Okay, so these are the different steps. First, we need to identify what is the gene is responsible for that enzyme. Okay, and we may also need to screen that gene from whole genome, or we need to identify the gene, okay, from the whole genome, or let's say from different databases which are present. The screening is the first step in order to identify, okay, this X gene is responsible for lysozyme or any Y or A enzyme. Okay, so first step is screening, and that screening could be from multiple sources like various databases, or if you don't have any databases present, then you need to find out the gene by yourself by different other methods. So first step is gene identification. Then the next step is gene cloning. So once we identify the gene, the next step is cloning, and the cloning is very complex procedure wherein you first need to amplify the gene. We need to have high copy number of the gene. We need to have multiple copies, and then we clone it into a specific vectors. We'll study about different vectors also in this class. Briefly, I'll give you outline. And then after cloning into suitable vectors, then we again screen whether our gene has got inserted into that vector. And once the vector is fully synthesized, where it includes our gene, the next step is that we overexpress that vector in order to get abundant protein. So that is what our aim is, to get abundant protein. Okay, so we need to do overexpressions. So in order to overexpress, first we need to transform it into our host cell. So host cells also may vary depending upon what type of gene you are looking for. The host cells could either be bacteria, they could either be yeast, and other eukaryotes like different insects. Okay, different insects and phages. So the host cells could be different number of mammalian cells and insects, bacteria, yeast. So depending upon the type of gene which we want to clone, there are multiple host cells. And then again, after transformation, we need to have a screening method in order to get that whether our gene is correctly inserted into the vector. And then we do analysis of that overexpression. Now the final step is the purification step. So this is our cell with a vector, and it will abundantly produce, let's say, this protein. Okay, and this is our vector, so it will abundantly produce some X protein which is of our interest. And now this protein will be present with other cellular proteins. Okay, so this protein will not be that pure because it will be present with the other cellular proteins in the host cell. So the last and final step is that we disrupt the cell. Okay, remove the cell fraction, remove different DNA, RNA, lipids, other macromolecules from the cell, and finally collect the protein fractions, which will contain all the cellular proteins, and it will include our abundant protein as well. And after collecting the protein fraction, we can do further purification by chromatography, HPLC, and then we can estimate how much protein we have got from doing all these steps. Okay, and then we can use our enzyme to do different reaction, to catalyze different reactions. So this is the overall general theme of today's lecture, that we'll work around these majorly these 4 steps only. Okay, so the gene identification, so we'll study each of these steps in detail. So the first step is gene identification and screening. So gene identification and screening, further again, as I mentioned, it could be divided into two. If you know the gene sequence, it's well and good, it's easier. But what if you don't know the gene sequence, and then you have to find out from the whole genome? So then you can either make a genomic library, or you can make a cDNA library. Now when do we go for genomic library? So genomic library is to be chosen when we are specifically targeting a particular sequence in the genome, whereas the cDNA library, as you can see here, it's basically when we target the transcriptome, that is the proteins which are expressed, basically expressed proteins in the cell. So if you are looking for if you are looking at transcription level, we'll go for the cDNA library, whereas if you are looking at the gene level, which are not captured in the mRNA or in the transcriptome, we'll go for genomic library. And then we'll screen those different sequences, we'll clone the sequences in the vector, and similarly if we know the gene sequence, again we'll amplify that gene sequence using PCR. We'll study about PCR also, and then with that amplified fragment, again we'll do the cloning. So once we get a fragment, the next downstream process for both the methods here is similar. Now what are the different steps in genomic library preparation? So the first step here, step 1 is, so the first step in the genomic library preparation is we first need to isolate the whole genome. So let's say we have a human genome. So first step is isolation. So here we have a human DNA. So isolation of genome is the first step. First we need to have the whole genome with us. The second step is then we need to do so we have a whole genome with us. So this is the first step. The second step is, this is EcoR1 is a restriction enzyme. We'll study in the future in the coming in today's class only what are restriction enzyme. Then the second step is RE digestion, that is restriction enzyme digestion. So we'll chop the whole genome. So this whole genome is of different approximately of different is of different size. So first we need to chop this by using restriction enzyme. Okay, and after chopping this, we'll get different fragments. Okay, different size fragments. Now these fragments will be of a specific length, and it's a randomly cut genome. So approximately we'll use a suitable restriction enzyme such that it will only chop, it will not chop within the gene, but it will chop just it will chop just outside our gene. If this is our gene, it will just chop outside the gene. Okay, flanking regions. So if these are the flanking regions of our gene, it will just chop there, and it's so it is very important that in a single fragment we at least have one gene. Okay, it is very important that we have one gene in the whole fragment. Okay, different fragments which we have, it will contain at least one gene. Now simultaneously we will also uh, have a restriction site cleavage point here, eco R1, and we'll simultaneously cleave the yak vector. Here we are using a yak vector. So for large genome size, we generally use yeast artificial chromosome because it packages a lot of DNA into it. So so it will package each fragment. Let's label this fragment A, B, C, D. It will package each fragment in these yak vector, and then we have finally we have a recombinant DNA. So these fragments will get inserted here. So let's say this is our fragment A. It will get inserted here, and then once it is inserted, the human, each gene is inserted into the DNA, we'll transform this vector, we'll transform our different vectors in our host cells. Okay, so so we'll do a transformation. So step 3 step 3 is to get this fragment and to purify this fragment. So step 3 is fragment purification. And step 4 is ligation. So this is the step 4 where you will ligate with vector. And then finally step 5, this is step 5 is transformation. Step 5 is transformation. So this is the general theme of cloning. Okay, restriction, digestion, fragment purification, then we'll ligate it with the vector, and then we'll transform the cells. Okay, so this is the step 5. So this is for genomic library, and then we can these are the detailed steps how to isolate genomic DNA. So as you can see, first you have to lyse the cells. So you can lyse it by sonication, or you can do lysis by different lysis buffers. Chemical lysis is also possible, and mechanical lysis is also possible. If you have tissues, you have to do homogenization and then lysis of cells. Then the second step is enzymatic digestion. So why do we digest it? Because the second step here is enzymatic digestion, because in order to have all the different molecules, in order to digest all the proteins, other macromolecule, we need to do enzymatic digestion. Then because after lysing we'll have different macromolecules like protein, we'll have protein, lipids, and other carbohydrates which we don't need. We just need our DNA, we just need our genomic DNA. So we need to digest all these other macromolecules by help of different enzymes. Then in order to just isolate DNA, we'll use absolute ethanol. So absolute ethanol will only precipitate out the DNA, and all the other macromolecules will be in the supernatant. Okay, they will be dissolved in the supernatant, only DNA will get precipitated. So here we'll have only DNA which is getting precipitated. Then by using phenol chloroform, what we'll do, in the aqueous phase we'll have DNA, and in the pellet we'll have remaining other protein and other macromolecules here in the green. Okay, the in the phenol chloroform purification, the DNA will get um, isolated in the aqueous phase. Then again we need to precipitate it to purify it more. Then we have a pure genomic DNA here. We have a pure genomic DNA finally with us. Okay, then next step is the RE digestion step. So as I mentioned earlier, we can do random digestion. We can do random digestion by different restriction enzyme, and we need to select different restriction enzymes. We can use a mixture of restriction enzyme also. So these are different standard procedures to use different type of restriction enzymes together. And the criteria to select the restriction enzyme is that we should have at least one gene fragment with us along with other flanking regions. Okay, we should have at least one gene fragment with us. Okay, otherwise it will defeat the purpose of RE digestion if it will digest the whole genome. So we'll have at least one DNA fragment with us. And we can also do mechanical digestion, but that process is again very random, and we sometimes might not get this important condition that we need to have at least one gene in the fragment. Okay, now so let's say our human genome is, or any animal genome is, approximately 2 into 10 to power 7 base pair long. Okay, so this is the average so this is the total size, not the average, total size of the genome. Okay, total size of genome is approximately 2 into 10 to power 7 bases, and we need average fragment length of 20 kb. So so we should have that information, that so approximately 15, if the gene size is 15 kb, average gene size across the whole genome, then keeping account of these flanking regions, we hope to have 20 kb of average fragment. Okay, so how many number of fragments we'll get? We'll get 2 into 10 to power 7 upon 20. So this is the average number of fragments. So this is number of fragments that we'll get after doing RE digestions. So this is around 10 to power 6. So we'll get 10 to power 6 fragments. Okay, so this is approximation that we use, that we to get. So if we get higher fragments, more than 10 to power 6, we can say that maybe it is not capturing one gene inside the fragment. Okay, maybe the gene is getting cleaved in between, that's why we are getting higher fragments, that's why we are getting more fragments. So this is just the approximation which we are using, that probably we'll get 10 to power 6 fragments. Then next step is to clone into a vector. So depending upon depending upon our gene size, we can clone it into different types of vector. As you can see, mycoplasma has very small size, so we cannot clone very large fragments, whereas in flowers or different amphibians, these vectors are in general designed in such a way that they can include different sizes, they can incorporate different huge sizes. Okay, So mostly we use in the gram negative or gram positive bacteria for cloning, or we also use yeast. Okay, so these are the approximate sizes which are used for cloning. uh, yeah, so the final step was transformation, right? So in transformation, what we do, we can transform this plasmid or into different host. Okay, so here are different host and how much insert it can accommodate. So these are the different vectors. We have plasmid, we have phage lambda vectors, we have cosmids, bacteriophage vectors, bacterial chromosome, yeast artificial chromosome. And as you can see, the yeast artificial chromosome has the highest capacity, whereas plasmids have lowest capacity because they generally get transformed into a bacteria. Now the next step was to prepare, the next method was to make cDNA library. So we had genomic library and we had cDNA library. So if we are interested in the transcriptomic levels, we'll go for cDNA library. So how to make cDNA library? Again, we need to have a transcriptome with us. So we'll have, so first we'll do, what we'll do, we'll isolate the mRNA. So we again, the same steps we are following. We'll lyse the cells, we'll purify the whole genome, we'll separate the DNA, we'll separate the RNA. uh, Usually DNA is separated by adding DNase enzyme, so all the DNA is getting cleaved off, so we are remaining only with the RNA. Then we'll isolate, collect the RNA. The next step here is, only different step is, we'll first use make cDNA, that is the complementary DNA. How we'll make complementary DNA? By using reverse transcriptase. So reverse transcriptase is the enzyme which uses RNA as a template, right? And it will, what it will do, it will carry out the first DNA strand onto mRNA. Okay, and this first strand which is synthesized on mRNA is known as cDNA. Why cDNA? Because it's a complementary DNA to our mRNA. Once we have DNA, what we'll do, we'll amplify that DNA. Okay, we'll amplify that DNA and then we'll insert it into our plasmids. So these are different plasmids. We'll insert it into our plasmids and then finally we'll transform them into different we'll transform them in the host cells and grow them, and then we can isolate different cDNA which we want, and we can do sequencing. Okay, the same step follows in the cDNA just as genomic DNA. Okay, so the step 1 here is mRNA isolation. So again it is following the same step. So mRNA has a unique structure. It has a poly A tail at 3' end. So at 3' end it has a poly A tail. Okay, and this poly A tail can be used for isolating this RNA. So what we'll do, after collecting our DNA and RNA together, what we'll use oligo DT beads. So these beads have oligo T attached to them. Okay, so these are the beads. It has oligo T, and these oligo T will be complementary to the poly A tail. Okay, and they will attach to these poly A tail here. Okay, and then we'll capture this mRNA, right? We can capture this mRNA using these beads, immobilized beads. Then finally we can just centrifuge this and we'll collect all the beads. We'll remove the supernatant, and then again in order to separate beads, we can do so we'll have beads with us. We can just denature this, then we'll be left with our mRNA. Okay, Very simple. After after collecting the beads, we can just denature these beads, separate the beads by just doing denaturation. We'll have a pure poly A RNA with us. Okay, we have purified mRNA. Then we have cDNA synthesis. So again, the cDNA synthesis is pretty much uh, same. So cDNA synthesis is of two types. So here this first type is homopolymer tailing. Okay, this is the homopolymer tailing method. In this method, what do we do? So this is our mRNA with a poly A tail here at 3' end. And so we use oligo DT primer. So we have this primer which is oligo DT. Okay, so this is the primer. Primer is nothing but it will help the transcriptase to find. Okay, it will help the transcriptase, reverse transcriptase with oligo DT primer, and it will transcribe the cDNA. So this is our cDNA, first strand synthesis. Then finally what we'll do, we have to get rid of this RNA. Then we'll use alkaline sucrose gradient. So RNA gets degraded in the alkaline condition. So alkaline conditions, RNA will get degraded because of 2' OH. Because of 2' OH on the sugar, there under alkaline conditions, RNA will get hydrolyzed. So we are only left with our cDNA here. Okay, we're left with our cDNA. Now after that, what we'll do, we'll add oligo G and reverse transcriptase. So we had a C poly C here at 3' end. We'll add oli G primer. Okay, and then we'll carry out second strand DNA synthesis. So we'll have a duplex DNA. Now this duplex DNA, we can do a PCR to get multiple fragments. Okay, we can do a PCR to get multiple fragments, or we can just add oligo G and oligo C primers to get multiple copies of the cDNA. Similarly, in this method is known as Guber-Hoffman method. So in this method, what do? We have this first cDNA synthesis step is same. When we have a mRNA and cDNA duplex, so this is our mRNA. So this is our mRNA and this is our cDNA. When we have this mRNA cDNA duplex, what we'll do, we'll add RNase H. So RNase H will cleave just the RNA. It will cleave the RNA randomly in between. Okay, so we'll have a small gaps, as you can see here. Okay, and then what we can add polymerases. Okay, we can add polymerases and we can add NTPs. Okay, so this polymerases will synthesize this strand, will synthesize the complementary strand by using these RNA as a primer. Okay, so they will synthesize the strand and they will merge, and then we'll have a duplex DNA. We'll have a double stranded DNA. Okay, because these NTPs will be all deoxy nucleotide NTPs. So we'll have a duplex DNA. So we studied, if the gene sequence is not known, we studied that we can make a genomic library or we can make a cDNA library, and from that we'll get our cloning fragment, and after that we have to do cloning. So when the gene sequence is known, we just simply have to do, in order to get multiple fragments, we just have to do polymerase chain reaction. So we can have a fragment with us, we can have a a gene fragment with us, we can simply do a PCR reaction. We can simply do a PCR reaction, okay, in order to get multiple fragments. Okay, in order to get multiple fragments, and then this fragment can be cloned. Okay, so just like uh, here, the final step for both is same, cloning. So yeah, so after getting fragments, before cloning, we have one more step, that is screening. So why screening is important? Screening is important in order to understand whether we have got the gene of our interest fragment, or we have got some other impurities. So screening is very important. So we have three types of we can do three types of screening. We can do DNA hybridization. This is very popular method, DNA hybridization. We can do immunogenic epitope screening. Immunogenic screening, wherein we use antibodies. We use antibodies against the protein which is synthesized, or we can use enzymatic activity. We can use the enzymatic activity of the protein, and we can see the we can add the substrate and check for the enzymatic activity. So so the first step is the DNA hybridization method. So this technique is known as replica plate technique. So this technique is very widely used for DNA hybridization. So we have a master plates here, and then we have different clones or different colonies, and this each colony is made up of one single cell, right? It is made up of one single cell which has our vector in it. Okay, it has our vector in it, and it will have our gene of interest. Okay, and it will have our gene of interest, and this gene of interest will make a protein, or it will make a enzyme that can be directly used in for screening. So for DNA hybridization, what we use a DNA. We use a probe DNA. As you can see here, we use a probe DNA that will go and bind to this our gene of interest. Okay, the probe DNA will bind to our gene of interest. Okay, the probe DNA. Why do we call it as probe DNA? Because this it has a probe which could be either fluorescently labeled probe, or it could be a chromophobe, or a chromophore also. Okay, so we have a DNA sequence that will get complementary. It is complementary to the gene of interest. It is very important. It will bind complementarily. It is complementary bound to a gene of interest. So what we'll do, so we'll have a master plate with us, and we have different clone, 1 2 3 4 5 6 7 different clone. Okay, so the first step is we'll take a nitrocellulose paper matrix, or a nitrocellulose paper. So why nitrocellulose paper? It has affinity for DNA. Okay, so this nitrocellulose paper will have affinity for DNA. So if we stick the nitrocellulose paper here, it will capture few cultures. Okay, it will capture our culture just like that, in the same fashion. Okay, 1 2 3 4 5 6 7. It will capture the same design from the master plate onto this microcellular paper. Okay, after this we'll lyse on this paper. We can just add the lysing solution, and we'll open up the cell, and will the DNA will be present, okay, in this paper, and the DNA will get bound to this nitrocellulose paper. The rest of the stuff we can just wash it out. Okay, now this bound DNA, okay, since it is a nitrocellulose, it will get bound to this paper. this bound DNA can be directly probed. Okay, this probes can be directly added onto this different colonies here. Okay, and then we have to just wait for them to get hybridized. Okay, so we can hybridize them at room temperature or at a specific temperature. They will get hybridized. As you can see, the seventh colony is hybridized, that is the probe here, it was showing some color or fluorescence. It was showing some color of fluorescence. Okay, so we identified that the gene of our interest is in the seventh clone. So then we'll go back to our master plate, and then we'll isolate this seventh colony from here. We'll isolate this seventh colony from the master plate. Okay, and then we can use this, and then we can label this, so that this seventh colony which we isolated, it belongs to gene X. So we can do that for multiple genes. However genes we have, we can do for this XYZ gene. Okay, same process is repeated n number of times till we get n number of genes which are required. So the probes could be of different types. It could be either made fluorescently labeled, or it could be radioactively labeled, and we have different methods in order to generate probes. So we have a random primer method, where we have a source DNA and we denature that DNA to get single stranded DNA. We'll separate the DNA. Denaturation means from double stranded we'll get a single stranded DNA. Okay, just like here we have a duplex DNA, here we'll get a single stranded DNA, and to this single stranded DNA now we'll attach have a label probe. Okay, which we'll just add radio labeled primers. Okay, we'll just add radio labeled primers and we'll synthesize small probes. Okay, we'll synthesize these probes. So the radio labeled nucleotide will synthesize this, and then we'll have a radio labeled primers like this. Okay, and these primers are used in the replica plate technique. Okay, these are the probes which are used for the replica plate technique. So instead of radio label, we can also use fluorescent label probes as well. Then the next method is terminal transferase method. This is also very important method, in which the terminal transferase is the enzyme. It will do, as the name suggests, at the terminal end, that is at the 3' end. Okay, it will attach one nucleotide. It will attach one nucleotide. Okay, so when if we have a duplex DNA like this, we can just add terminal transferase and whatever nucleotide, radio labeled nucleotide we want to have the probe with. So if we want ATP to be radio label, we'll just add radio label ATP, and then we'll have at 3' end we'll have a radio labeled ATP, or it could be fluorescently labeled as well. Okay, so these are the two methods by which we can make different labeled probes. Then we had 3 steps. We had DNA hybridization. Second one was using immunogenic, that is antibodies, and the third step was using enzymatic activity. So it follows the same principles of replica plating, where you will be having a master plate. You will using some matrix. In case of DNA, you will use nitrocellulose membrane. In case of other, you can use PVDF membrane also, or other Wattman filter paper. So there are different membranes which can be used. So this membranes or matrix will attach our colonies. Cells will get transferred. Small amount of cells only will get transferred, and this after that will lyse the cells, open up, and there will be a bound, so that those cells will have a mixture. It will have DNA, it will also have protein. Okay, it will also have protein. So if we have antibody against these proteins, okay, so if we have our abundant protein which we have overexpressed, and we have antibody against them, what we can do, we can just add antibody. We can add primary antibody, and then we can add secondary antibody to the same matrix. So the secondary antibody will go and bind to the primary antibody like this, right? And this secondary antibody, this is our primary antibody. The secondary antibody has again it has a probe, which could either be a fluorescent, it could be a fluorescent, or it could be a chromogenic substrate also. Okay, then once we add then we can observe it under we can see the fluorescence, or we can either see the color develop. Okay, so these cells which are bound, okay, they are showing a color, which means that this colony here is basically expressing the abundant protein. So we are screening them. Okay, we are screening them from n number of cells. So these are the screening methods, right? So we are screening them from this n number of cells, that only this colony or this cells have our X protein. Okay, they are expressing our protein. Okay, other cells are just false positive or present there. Okay, similarly in the enzymatic method, instead of primary antibody or secondary antibody, you add enzyme. Okay, so this enzyme will react with that substrate, this enzyme will react with that protein, and it will give you some color. Okay, same principle for both. So again we we have done the we saw how to do screening, and then how to get the clone of our interest. Okay, we have found out how to get the clone which has the fragment in it, and the final step is the cloning. Okay, and here also after getting the fragment, we have a cloning step. But how do we get this fragment in the sequence which is known by PCR reactions? Okay, so so this is the basic concept of PCR reaction, that we have a duplex DNA. Okay, so we have a initial DNA, we call it initial DNA. Okay, which we used it so initial DNA which we want to amplify. So let's say this is our gene of interest. Okay, this is our gene fragment which we will amplify, which we want to amplify, and further next step is cloning. So the first step in the PCR is denaturation, that it will separate this duplex DNA. It will get separated into single stranded, as you can see, red and blue, it both are separated. Then we have primer binding to each sides. Then we have first cycle, we have first strand synthesis, and then finally exponentially it will have different synthesis of strands. So what are the different steps here? So the first step, initiation, is where we denature our duplex DNA. So that is why we are using very high temperature. So this is the temperature shown here. So we are using around 90 to 95 degrees Celsius. Okay, and this initiation generally is known as hot start. Okay, because why we are using very high temperatures. And then denaturing temperatures are generally at 94 to 96 degrees Celsius. At this temperature, almost all the duplex DNA will get converted to single stranded DNA. Okay, sorry, will get converted to single stranded DNA as shown here. Okay, then after denaturation, okay, the first step is initiation, where we start with heating up at 5 minutes. So this is time, and this is temperature. Okay, this is time here, and this is temperature. So the first step is we initiate to heat at 5 minutes at 95 degrees Celsius, and Then along with this step, we also the denaturation step um, is combined with this step, or along with hot start we have denaturation step combined. Okay, so the denaturation step lasts for 30 seconds. Okay, so the denaturation step, so we have uh, these are the major steps. Denaturation, which is around 95 degrees Celsius, 94 to 96 degrees Celsius, and it will last for how many? It will last for 30 seconds. Okay, it will last for 30 seconds. Then we have annealing. So what is annealing? The primers will come and it will get attached to the two single stranded DNA. The second step is annealing, where the primers will come and it will get bind to these single stranded. So this is the annealing step, where the primers are coming and they are getting bound to the single stranded DNA. Okay, and this annealing step takes place at 56 degrees Celsius. Okay, approximately the annealing temperature is from 45 to 60 degrees Celsius. Okay, this is the annealing. um, uh, Generally we don't go till 60, we are it is around 45 to 58. Okay, this is the annealing temperature. um, and this temperature is for 45 seconds. Okay, it lasts for 45 seconds. So this is not hard and fast rule that we should use only these temperature for this particular time period. You can change it according to your template or according to your gene. Okay, so these optimization is necessary for according to the gene of interest. Okay, whatever gene that you have, you can optimize the step. Okay, then third step is elongation, where now you have primers attached to the single stranded DNA, and these primers now they will have a they will elongate the new strand. Okay, they will elongate the new strand, and this process takes place at 72 degrees Celsius with help of Taq polymerase. Okay, and this process will last for 2 minutes 30 seconds. Okay, it will last for 2 minutes and 30 seconds. So or it will last for 2 minutes to 5 minutes. Okay, elongation step, it will last for 2 minutes to 5 minutes, again depending upon how long your fragment is, the time you can change the time. Okay, now this stages will increase depending upon how many cycles you have. If you require n cycle, you will so we generally keep the cycles between 20 to 30 cycles. At 20 to 30 cycles we'll get our product. Beyond 30 cycles, even the polymerase stops working and you will get different it will add wrong nucleotides. The efficiency of the polymerase will reduce, so we don't go beyond 30 cycles. So 20 to 30 cycles is the limit. So again the cycle will after elongation it will go back to denaturation step. That is, after elongation you will have a duplex DNA again. Okay, we'll have a duplex DNA again. This will further get, it will again go back to step 1. Okay, it will further get denatured at 95 degrees Celsius. Okay, this step will go back here, it will further get denatured at 95 degrees Celsius. Now we'll have 4 single stranded strands here, this red, yellow and blue and red or pink. Now this 4 single strand, again the primers will come and bind to those strands, and there will be again annealing. Okay, annealing step, elongation step, and then this cycle continues up to 30 cycles. Okay, max 30 cycles. And after the end of 30 cycles you'll get your PCR product. Okay, so this is the PCR polymerase chain reaction. And finally, once you get the PCR product, what you will do? You will do cloning. So this is the general cloning slide, very compact steps shown here, but it is it has much more more tedious steps to it. So we have either genome or we have a cDNA with us at this side, where we get different genes by using different vectors. We get it into different gene, then we cut the gene, okay, by using some RE. Similarly, we cut the same use the same RE here to cut our vector also, and then we ligate this. So this is a gene of interest which is a ligated into vector, then we transform it and then we get clones of the enzyme which we are interested. Okay, this is the general cloning procedure which is used for enzyme production. So we'll go details into these restriction enzymes. Okay, what different enzymes which are used for cloning. So restriction enzymes, as you can see, they are isolated from bacteria. So why does bacteria have restriction enzymes? Because they are used for their own defense system. So whenever the phage attacks bacteria, it integrates its genome into bacteria. So in order to recognize the bacteria self DNA, so we call it as self DNA. So in order for the bacteria to recognize its own DNA or self DNA, it methylates its own DNA. Okay, and at this methylation, so this methylation protects the restriction enzyme digestion. Okay, so whenever there is methylation on the DNA, the restriction enzyme won't cleave those sites. Okay, thus the bacteria will protect its own DNA, and the phage doesn't have any methylation on its genome. So the phage DNA will be unmethylated, right? The phage DNA will be unmethylated, and that will be cleaved by restriction enzyme. So this is how restriction enzymes are used as a self defense for bacteria. So we have isolated this restriction enzyme because they are very unique, and they are very widely used for cloning purposes, right? Because they give us, so they cleave at specific site. That this is very important. They cleave at very specific site, and they will give us sticky ends. Okay, so they will give us sticky ends, and this sticky ends can be further used for cloning it into different vectors. Okay, so the sticky ends can further get complementary attached to itself or other DNA fragments. Okay, so as you can see here, there is no methylation here, whereas we have a methylation here. Okay, there is a methylation here. So because of this methylation, this DNA is not degraded, whereas there was no methylation here, so the DNA gets degraded. So this is the basic strategy of restriction enzyme, and as you can see, they produce sticky ends. So these sticky ends can get complementary attached to other sticky ends or by itself. So this property is very vastly used in cloning purposes. Okay, so I'll just remove this. Yeah, so we have different types of restriction enzyme: type 1, type 2, and type 3. So when these restriction enzymes were isolated, they had different properties, and depending on those properties, they were divided into different types of restriction enzymes. So we have type 1. What it does, it will recognize this sequence. So it will recognize a palindromic sequence. So what is palindromic sequence? It reads same from opposite ends. Okay, from opposite ends it reads same, that is GAATTC from opposite end it will also read as GAATTC. Okay, so it will recognize. Uh, so type 1 restriction enzyme, as you can see, the recognition side is non-palindromic. It doesn't recognize any palindromic sequence, and the cutting site is 1000 base pair away from the site. So this property is not that great to be used in cloning because it will cleave something else other than our gene. So this is not that great to be utilized. Um, so these are the different types of type 1 restriction enzymes. Then again, type 3 is also similar. It doesn't recognize the palindromic sequence, and again it cleaves 20-25 base away from this recognition site. So again, this is not suitable for cloning, but type 2, as you can see, they recognize the palindromic sequence such as this. Okay, and they cut within the recognition sequence, so they cut like this and give sticky ends. Okay, sometimes they could be sticky, sometimes it could be blunt. By blunt I mean instead of sticky ends like this, they could be just blunt cutting. Okay, just like this without any sticky ends. This cut is known as blunt cut. Okay, and so it recognizes the palindromic and it cuts within the recognition site. So this is very important characteristic of restriction type 2 restriction enzyme, that it will cut within the restriction site. Okay, and it will give a sticky as well as blunt end, and these are the cofactors which are required for the enzymes. The next important enzyme is the ligase enzyme. So what does this ligase enzyme do? As the name suggests, ligase, ligase is joining two DNA strands. Okay, so we have so whenever there is a new strand, so if we have a primer here, okay, and we have a primer here, okay, on a replication process, so the new strand will be synthesized here and it will stop. Similarly, again it will begin and it will stop. Okay, now this primer is cleaved off. Okay, this primer is cleaved off and it will continue by DNA polymerase. And what will happen? There will be a nick. There will be a nick here. Okay. Why? Because it cannot join this. It cannot join the phosphate and OH. 5 prime phosphate and 3 prime OH cannot be joined by by DNA polymerase. So there is a nick here, and this nick is very harmful. So DNA ligase, what does it do? It creates a phosphodiester bond. Okay, it creates a phosphodiester bond. So ligase is part of synthetase enzymes or synthase enzyme. So whenever there is a formation of bond, there will be require of energy. So either it utilizes ATP or NAD+. Okay, so we have two ddNA ligase which are used in molecular cloning, that is T4 DNA ligase and E. coli DNA ligase. So this T4 DNA ligase, what does it do? It utilizes ATP, whereas E. coli DNA ligase, it utilizes NAD+ as a source of energy. Okay, and after utilizing this, they create a phosphodiester bond. So this nick is repaired. Okay, this nick is repaired, or this nick is ligated. So we'll have a duplex DNA without any nick. Okay, so this is the purpose of ligases, very important in normal replication processes also, and as well as in the cloning. Then we have alkaline phosphatases. These are also very important enzymes. So what does alkaline phosphatase do? So we have so this is one strand, okay, with 5 prime phosphate, and this is the complementary strand with the 3 prime OH. Okay, these are the two strands. This is a duplex DNA. Then we have a 3 prime OH and 5 prime phosphate strand. So So the ligase, what ligase molecule will do, it will form a phosphodiester bond between this, and it will ligate into a completely duplex structure. Okay, it will ligate into completely duplex structure just like this. Okay, without any nick. Okay, now what alkaline phosphate does is it will remove the phosphate from the 5' end. It will remove this phosphate. Okay, and it will have a hydroxyl group here. So what will happen if you add ligase? There won't be any ligation. Okay, there won't be any ligation. So why do we do this? Suppose if we have a vector here like this, and we cut it by restriction enzyme. So we cut this fragment. Okay, we cut this fragment using restriction enzyme, and this fragment is lost. And this fragment here will have a gene of interest ligated into this. This is the gene of interest, let's say. This is the gene of interest which we want to be ligated here in between the red duplex DNA. Okay, this should be as seen here. So if we add ligase, there would be a possibility that the vector itself can get ligated back just like this. Okay, the vector can itself get ligated back without our gene of interest going inside this. Okay, without our gene of interest going inside this. So this is why we use this alkaline phosphatases. So the 5' end will be completely removed. The 5' phosphate is removed so that there won't be self ligation of the vector. Okay, to avoid, this is very important, to avoid self ligation of vector. Okay, then the gene of interest will come. Then we can further hydrolyze this, and we can ligate it by the gene of interest. Okay, so this is why alkaline phosphatases are used. So again, so what are the different vector properties? So there are different types of vectors: bacterial vectors and yeast vectors and eukaryotic vectors. So what are the ideal properties that we must have? So the property one is it should be low molecular weight. For example, this is a vector. Okay, so it should have a low molecular weight itself. So it has multiple components. So what are the different components of vector? These are general components. We have a origin. As you can see, this is the origin. Okay, this is origin 1. We have origin. Then we have different selection markers. So selection markers here are ampicillin and tetracycline. Okay, this is PBR 322. Okay, and what are other components? Then we have, for eukaryotic, we have promoters also. And then different other selection markers also for blue-white screening, if you guys know what is blue-white screening. So we have screening markers and we have selection markers also. So blue-white screening is for screening markers. Okay, it's different than selection markers. Okay, so what are the properties of vectors? That it should contain all this component and it should be low molecular weight. Okay, it should not be very high molecular weight, so that it might not reduce our gene of interest capacity. Okay, so we need to also put our gene of interest, and if the vector is itself is of high molecular weight, then the whole plasmid will be of high molecular weight, so that will restrict the gene fragment size that we want to put in. So it should be a low molecular weight, then it should have easy transformation, it should be easily transformed into the host cells. And the third property is we need to see some phenotypic changes like some color changes after successful vector insertion. We should see some color changes or some properties like ampicillin resistance or tetracycline resistant by this screening selection markers and screening markers. And the fourth property is that it should have multiple restriction sites. It should have multiple restriction sites. As you can see in PBR 322, we have multiple restriction sites. Okay, you can use n number of restriction sites. Okay, for the cloning purposes, these are all the restriction sites where you can insert your gene of interest. Okay, and the fifth and most important is it should have a high copy number. What do you mean high copy number? So copy number is the number of number of units of plasmids which are present in the vector. So it should have a high copy number in order to give us multiple in order to give us a higher protein at the end. Okay, so these are the properties and components of different vectors, and this is PBR 322 with ampicillin and tetracycline resistant gene selection markers. So this is the step for plasmid isolation. So it is a bit different from genomic isolation. So we studied that we do lysis to isolate the genome. So we'll have both chromosomal DNA as well as plasmid, which is very small molecular weight as compared to the chromosomal DNA. Okay, then what we'll do, so here we are separating these based upon different sizes. So we are utilizing this size fraction here. So we actually are able to precipitate the huge chromosomal DNA using different reagents, and the plasmid DNA along with the protein gets remained in the supernatant. Okay, and this plasmid can be further purified, and all the protein which are present can be removed by phenol chloroform separation, and then we'll get a pure plasmid with us. Then further we can purify this plasmid by doing alcohol precipitation. Okay, so we'll have this pure plasmid with us. There are the eukaryotic plasmid like YP24. It's a yeast plasmid, as you can see, and again it has origin. So this YP plasmid, it's a shuttle vector, that is it can also be used in bacteria as well as yeast. So it has origin sites for bacteria for its replication, as well as it has origin sites for the yeast replication as well. Okay, then we have yeast artificial chromosome, which we studied in the during cDNA or genomic library preparation, that yeast artificial chromosome can pack huge amount of DNAs in MBs. So large chunk of DNAs can be packed into these vectors. So these are used for genomic library preparation or cDNA library preparation. Okay, then we have different phage plasmids. Okay, these are also very important. So phage plasmids have just like other plasmid, it has different sites. Okay, as shown here, and these different sites are responsible for making the packaging material. So what is this packaging material? So this packaging material are the head and tail of the phages. So it has multiple sites on the phage plasmid, and they synthesize these packaging material, and these packaging material are further when made, like head here, they will incorporate these plasmids. Okay, so here at the end region we have cos sites. Okay, these cos sites, okay, on the end region we have cos sites. The cos sites are pinched here, so these are the cos sites. So these are the end sites uh, of the phage of the whole genome, that will they will pinch, and they will only these sites, only the size on the DNA which are included in this cos site will get packaged into the phage. Okay, and then this packaged DNA further could be further used to infect other bacteria. Okay, so the phages infect different bacteria. So now it has packaged this DNA, and we can also insert our DNA here, or gene of interest here. Okay, so this is a gene of interested. So this will get packed, this whole genome will get packed in phage, and when it will infect different DNA, so we there this here we won't have any transformation, we'll have phage infection. So after infection of this phage onto different or host cells, which are mostly bacteria, the phage will, what it will do, it will transfer our this DNA, packaged DNA, into bacteria. Okay, it will package it will put the DNA into bacteria, and then this DNA can be integrated into the bacterial genome. So we'll have either integrated plasmid, or we'll have a free plasmid, free extra chromosomal free plasmid. It's also known as extra chromosomal. Okay, so these are different vectors, and with this I conclude my slides, and we'll move on to assignment questions. So I request you guys to answer different assignment questions. Okay, so these are very simple. Okay, I'll open my chat box, you guys can even answer in chat if you want. Or you can unmute yourself also. So the first assignment question is the number of DNA duplexes from one DNA duplex, that is one double stranded DNA, after 4 cycles of PCR is 8, 4, 32 or 16. So anybody knows how to calculate this? Yes? Anybody knows how to calculate this? You have to simply use 2 raised to n formula. 2 raised to n, why? Because every time there will be a duplication of the fragment, that's why it will rise exponentially as 2 raised to n. So n, what is n? N is number of cycles here. N is number of cycles. Okay, n is number of cycles, and So the roughly after 4 cycles, if we have a duplex DNA, how much we'll get? Yes, so we'll get 16 2 raised to 4, that is 16. Okay, so the correct answer is 16. Everybody got this? Is it clear for everybody? You can answer in chat, or you can just unmute yourself. So the next question is, primers used for the process of PCR are single stranded RNA oligonucleotide, single stranded DNA oligonucleotide, double stranded RNA oligonucleotide, or double stranded DNA nucleotide. So what is the answer here? Primers are supposed to be single stranded so that they can anneal to our single stranded. Yes, the answer is B, single stranded DNA oligonucleotide. Okay, we don't use RNA primers because Taq polymerase, so RNA primers, for one reason, they are very pretty much unstable. RNA is less stable than DNA, and DNA primers are much more uh, efficient than RNA primers, so we used DNA primers. Okay. The most effective technique for enzymatic amplification of specific gene fragments of DNA is DNA hybridization, nucleotide sequencing, polymerase chain reaction, and reverse transcriptase. So hybridization again is it's just getting hybridization. There is no enzymatic amplification or anything. Nucleotide sequencing, again there is no amplification. Polymerase chain reaction, yes, we do amplify. Reverse transcription, again it will just transcribe cDNA. Okay, reverse transcription is not amplification, it is just first strand synthesis. So the answer is C, PCR. The enzyme that creates sticky ends during homopolymer tailing is one of the method used for cDNA synthesis, right? So homopolymer tailing, so alkaline phosphatase, what it will do? Alkaline phosphatase will remove 5' phosphate. It will 5' phosphate removal. So by removing 5' phosphate, we cannot have sticky ends. Then we have polynucleotide kinase. Polynucleotide kinase is just it will do reverse of what is alkaline phosphatase, that is it will add 5' phosphate, 5' phosphate addition. Okay, DNA polymerases, what does it do? DNA polymerases, it will just synthesize the duplex strand. Okay, it will synthesize the duplex strand. Then we have terminal deoxy nucleotide transferases. So what is terminal? As the name suggests, it will add ribonucleotides or deoxyribonucleotides at the end. So if we add NTPs, if we had ATPs, let's say, with this enzyme terminal dinucleotide transferase, it will add a poly A tail. Okay, and it will generate a sticky end during homopolymer tailing. So the answer is D. Okay, the answer is not C, the answer is D. Ligation takes place between adapter and linker and vector, 5' terminus and 3' terminus of terminus and adapter and linker. So the ligation takes place, as I mentioned, it takes place at 5 prime phosphate and 3 prime OH. Okay, the ligation reaction takes place between 5 prime phosphate and 3 prime OH. Okay, the answer is C. So adapter linker are different concepts. So linkers are nothing but chunks of duplex DNA or small fragments of linker DNA which are added to the gene of interest. Okay, if this is our gene of interest, okay, this is a gene of interest, let's say GOI. Linkers are nothing but small additions onto the gene of interest. So these are linkers. Okay, and what are adapters? So adapters are kind of linkers only, but they have what they have a restriction site. Okay, they have a restriction site on them, that's why where we can generate a sticky end. So they have a restriction sites on them. Okay, so only difference between adapters and linkers are adapters have restriction sites on them. So the correct answer is C. DNA ligase enzyme used in genetic engineering. So we used in genetic engineering we use T4 DNA ligase. Okay, we generally use this T4 DNA ligase. Okay, so the correct answer is T4 DNA ligase. So this ligase again, it will 5 prime phosphate and 3 prime OH. It will ligate these two molecule. Okay, so we use T4 DNA ligase and E. coli ligase enzyme. DNA ligase mediated reaction during DNA replication. So DNA ligase, as I mentioned earlier, so we have so during replication processes, what we have are a template strand, and onto this template we have a primer. Okay, so we have a multiple primers attached. Okay, so this is on the lagging strand. So on the these are the RNA primers, right? These are the RNA primers which are attached, and then we have synthesis of strands. Okay, we have a synthesis of strand in this direction. So after removing of primers, we'll have small gaps. Okay, and these fragments are known as Okazaki fragments, right? These fragments are known as Okazaki fragments, and there will be a nick here, and this nick is filled by DNA ligase enzymes. So formation of a phosphodiester bond, this nick is filled by phosphodiester bond formation between 3 prime OH and 5 prime OH on the lagging 5 prime phosphate, actually on the next lagging strand. Okay, the RNA strand in RNA DNA hybrid is removed by, so we can remove that RNA by RNase H. Okay, the answer is B, RNase H. We can remove the strand by RNase H addition. So RNase is again it will just chop all the RNA. Okay, it will just chop all the RNA. It will not just it will chop the double stranded RNA mostly, or single stranded RNA. RNase H is it will remove it will recognize DNA RNA hybrid. This is very important. It will recognize DNA RNA hybrid, and it will remove this. It will chop off only RNA. Okay, that's why RNAs cannot be used. RNAs cannot recognize DNA RNA hybrid. Nucleases Again, it is very random. It will cleave, nucleases will cleave both DNA and RNA. Phosphatases, again, no use. It will not remove DNA RNA hybrid. The correct answer is RNA is H. The dash endonuclease cleaves both single stranded and double stranded DNA molecule in non-specific manner. So anybody knows the answer for this? Which endonuclease cleaves both single stranded and double stranded DNA in non-specific manner? Yes. Yes, you guys can answer. So does DNA's can DNA's cleave both single stranded and double stranded DNA molecules, or will it just cleave double stranded DNA molecule? Yes. So there are two things here, endonucleases and exonucleases. So what are endonucleases? As the name suggests, endonucleases are ones which will cleave in between. Okay, they will just cleave in between. Okay, and exonucleases, again exonucleases, they will chop off the DNA from the ends. Okay, they will start chopping off from the ends. Okay, so here the question is, which endonucleases both cleave single stranded and double stranded DNA molecule in non-specific manner? So first, so this is the restriction enzyme. This is the restriction enzyme. So they will not cleave. S1. So what does S1 endonucleases? S1 is the endonuclease. What will it do? It will break down single stranded DNA and RNA. It is very important, it will only break down single stranded DNA and single stranded RNA. But DNA is one. Okay, DNA is one, it will break down both single stranded as well as double stranded DNA molecules. Okay, so the correct answer is DNA is one. The following types of enzyme responsible for removing individual nucleotides from the end. So again, as I mentioned in the previous slide, so exonucleases, they will chop off from the end. Exonucleases, they will chop off the nucleotides from the end. So ligases, they won't do any breakdown, they will just ligate the two molecules, right? Exonucleases is the correct answer. Endonucleases, again they will break off in between. Modifying enzymes, no. So the correct answer is exonucleases, B. The type of bonding present between N1 pyridine and deoxyribose is of DNA is dash. So we have pyrimidine ring and we have pyridine ring. So pyridine is a 9-membered ring like this. Okay, and we have a N1 here. Okay, this is the N1 of. Okay, so this is pyrimidine, and pyridine is. Sorry, this is pyridine and this is pyrimidine. And this is. Okay, so N1 forms a glycosidic bond with sugar. Okay, CH2OH, we have a CH2OH and it forms a N glycosidic bond. Okay, it forms a N glycosidic bond. So it's as it is with deoxy sugar, it forms a N glycosidic bond. Phosphodiester bond is formed between the phosphate group and the 3' hydroxyl group. 3' hydroxyl group on the sugar, which is here. Okay, so this is the 3' hydroxyl group, and the phosphate group. This phosphodiester bond form is between this. So the answer is N glycosidic bond. The difference between sugar molecule of DNA and RNA is again very simple. So what are the differences? So in sugar molecule, so this is the pentose sugar molecule. So this is deoxy, let's say this is deoxy, 2-deoxy, that is the 2' OH is missing. Okay, the 2' OH here is missing, that's why 2-deoxy. Whereas in ribose, what do we have? We have a 2' hydroxyl group present here. Okay, HOH. So this is the ribose. So in ribose you do have a 2' hydroxyl group, this is present, whereas in deoxy you don't have 2' hydroxyl group. So the correct answer is absence of oxygen on at 2C. So this is the second carbon, so the oxygen is absent. Polymerase utilize PCR is extracted from. So since it has to act elongate at higher temperatures, that is the elongation temperature is optimally at 72 degrees Celsius, and thus this Thermus aquaticus bacteria, it's a thermophile, it's a thermophilic bacteria. Okay, that's why the name Taq polymerase. So this Taq is considered from this T and AQ, Taq Thermus aquaticus. So we use Taq polymerase to in PCR, and it works very nicely at 72 degrees Celsius since it's a thermophile. That is, it is present in different thermophilic environment. Then antibiotic resistance present in PBR 322 is, so there are two antibiotic resistance screening genes, selection genes present. One is tetracycline and the other is ampicillin. Okay, so both the answers are correct, tetracycline and ampicillin. The enzyme which separates two strands during DNA replication are gyrase, topoisomerases, helicase, and DNA polymerases. So the two strands are separated using helicase. Very simple. So gyrase and topoisomerase are decoiling enzyme. Okay, they will release the tension in the DNA polymerase is a synthesizing enzyme. It will synthesize the DNA with the help of a template. So helicase is the only one which will separate the two strands. So this is the end of the class. Any questions? Yes, any questions? So if you don't have any question, I'll stop the class for today. Okay, thank you everyone.